Hello viewers of Biotecnika, welcome back again. Today I'll be talking about CSIR NET Exam 2022 morning shift answer key explanation, very specifically in unit number 6, System Physiology in Plants. So let's talk about it, and I believe that all of you might have done your CSIR NET examination really well, and let's talk about all the answer key explanation in detail. So the first question, at which of the following electron transport chain complexes does antimycin A typically inhibits the respiratory chain? Yes, we know ETC is otherwise called as respiratory chain because almost 70% of the oxygen is utilized here, which means oxygen is the final electron acceptor. Now the question for us is, antimycin will inhibit which complex? It's a direct question. So the answer is complex number 3. So we'll understand how this actually comes. So we know that this respiratory chain or ETC actually main purpose is to convert NADH or FADH, which is the indirect source of energy, into the direct source of energy, which is ATP. Which means we have a check in hand, we have to convert them into a cash. And this process has to be done through electron transport chain. The names gives an idea that there is actually a transfer of electrons. Electrons are going to be transferred from low redox potential or negative redox potential to a positive redox potential. Redox potential to a positive redox potential or a high redox potential. And this electron transfer is actually coupled by proton translocation from the matrix to the intermembrane spaces. Suppose this is going to be the mitochondria, which is the inner membrane. So ETC takes place in the inner membranes of mitochondria. So this is the inner membrane, and we know inner membranes are going to be lipid soluble. And then there's going to be an outer membrane which is present here. So let me consider this red part is going to be the outer membrane. So this is going to be the outer membrane, and the space between the outer membrane and the inner membrane is called intermembrane space. Intermembrane space. And usually all the complexes are embedded in the inner membranes. All the complexes like complex number 1, complex number 2, complex number 3, all are actually present here. So let me make complex number 1. So this is going to be complex number 1. And this complex number 1 is called as NADH dehydrogenase ubiquinone oxidoreductases. Ubiquinone oxidoreductases. And the second complex is called as succinate dehydrogenase. So the second complex is going to be succinate dehydrogenase. This is succinate dehydrogenase ubiquinone oxidoreductases. And the third complex, the question is about the third complex, which means antimycin. So this complex is called cytochrome C ubiquinone oxidoreductases. And this is otherwise called as cytochrome BC1 complex. And the fourth complex is called as cytochrome AA3 complex, or it is called as cytochrome C oxidases, not oxidoreductases, it is oxidases. We know that every enzyme which is embedded in the membrane, which means they are all lipid soluble, here they have electron carriers, which is FMN and FES. These are all lipid soluble, whereas ubiquinone, or we can call it coenzyme Q, or water soluble. That's why they are not embedded here. In complex number 2, they have FES as an electron carrier, and in complex number 3, it is called cytochrome B and cytochrome C1. That's why it is called a cytochrome BC1 complex. In between, they have FES. Very simple. And in this one, they have cytochrome A and A3. Now the question for us is antimycin A. There is an inhibitor which is called as antimycin A. So if anti-mysin A is present, which of these complexes are actually inhibited? Yes, we know it's definitely going to be complex number 3, which is cytochrome C ubiquinone oxidoreductases. What will happen is the electron used to flow from NADH. NADH usually enters this complex number 1 by losing its electrons, and FMN will get reduced. Now the electron will be transferred from here and it will be keep on going, because as the electron goes, the protons are actually pumped from the matrix to the intermembrane space, all of you know. So now what exactly happened? If anti-mysin is present, they will not allow the electrons to go to this one. If anti-mysin is present, they will not allow the electron transfer from cytochrome B to cytochrome C1. So we can say it blocks the electron flow from cytochrome B to cytochrome C1. So we know that it's definitely going to be complex number 3. So the correct answer for this question is going to be option C. Okay, let's talk about the other inhibitors which inhibits all other ones. So let's talk about it. So the inhibitors, complex number 1 we'll write, complex number 2, complex number 3 and complex number 4. So the complex number 1 inhibitors, we will write the names. The complex number 1 inhibitor, you can just have mnemonics like roti and non. So remember rotinons. There is rotinone which inhibits complex number 1, NADH dehydrogenase, and there is other thing called as amoborbitol, and the second one is going to be pyrisidine. This is also the inhibitor. So this is actually a direct question which has come, which is anti-mysin. So the second one we know it is succinate dehydrogenase. So definitely it is malate. So malate is an inhibitor. And then they have carboxin is an inhibitor. And then they have TTFA. And the third, just now we have seen, it's definitely going to be anti-mysin. So anti-mysin A. And then there's going to be dimercaprol. And the fourth, all of you know, it's definitely going to be cyanide and hydrogen sulfide and cobalt. So this, if you know this chart, definitely you would have answered this question. So the correct answer is definitely going to be option number 3, complex number 3, anti-mysin A blocks electron flow from cytochrome B to cytochrome C1. Yes. Now let's go to the second question. Question ID 20, and it's part B question only. Very simple question. If you know the answer for chlorophyll, if you have known the structure of a chlorophyll, then this question is definitely going to be easy. And we know that chlorophyll A and chlorophyll B, C, D are also present, but here the question talks about chlorophyll A, and the question is which of the following statements regarding chlorophyll is not correct. We have to find out which of the statement is not correct. So we know that there is chlorophyll A and chlorophyll B. And chlorophyll A R group is definitely going to be methyl group. And chlorophyll B R group is definitely going to be aldehyde group. So let's check for the statement. Chlorophyll A has a methyl group in its porphyrin ring, correct? So this is a correct statement, so we cannot choose it. The chlorophyll B has aldehyde group in its porphyrin-like ring structure, yes. And whether it is chlorophyll A or whether it is chlorophyll B, they will definitely have a magnesium ion in the center of the porphyrin ring. But let's check the statement number 3. Only chlorophyll but not chlorophyll B has magnesium, which means they are telling chlorophyll A is having magnesium and chlorophyll B does not have magnesium. Yes, it's not the case. Both has magnesium. So this is the wrong statement. So I will be choosing this option is the incorrect statement. Let's check anyway for the last statement. The long hydrocarbon chain, which is 20 carbon, it is hydrophobic. And that's the reason, because of this hydrophobicity, they are actually present in the thylakoid membrane, photosynthetic membrane, which is thylakoid membrane. So this is definitely going to be a correct statement. But they have asked which is the not correct statement. So definitely it's statement number 3. Now let's look at the structure. We have a porphyrin head, cyclic tetrapyrrole structure, which is responsible for light absorption. So this is the head. You can literally see here. This is actually the head. And this is the tail, which is a hydrophobic tail. And here you are seeing a pyrrole ring. And this is a linear pyrrole and it's tetrapyrrole, but it's linear here. Here you are seeing a cyclic one. And if R group is present, methyl group, then it is chlorophyll A. If aldehyde group is present, it is chlorophyll B. And whether it is chlorophyll A or chlorophyll B, they definitely have a magnesium ion. So the statement goes wrong in the third one. And whereas this phytol ring is hydrophobic in nature, they are embedded in the thylakoid membrane because of its hydrophobic nature. So the correct answer for this question is option number 3. Okay. Now let's move on to the third question, which is question ID 81, part C question. So let's talk about this question. Glycolysis and citric acid cycle contributes precursor to many biosynthetic pathway in plants. Yes. So first let's check. If you know the first one, you can definitely end up with the correct answer. So the first one is going to be hexose phosphate. So let's make it like hexose means a 6 carbon sugar. So hexose phosphate is going to be a precursor. So here they are talking about the precursors. And here the products. So from a precursor, what are the products that is responsible for producing some other things in the plant will be done. So hexose phosphate. So 6 carbon sugar means definitely we would be knowing it's going to be glucose. It might be a glucose or fructose. For now we can make it as glucose. Glucose is going to give which of these things? Definitely cellulose is a carbohydrate. And it's going to be a polysaccharide, definitely a structural polysaccharide. It is made up of beta-D-glucoses. So many beta-D-glucoses make up cellulose. So if you know the first option, definitely you would have come up with a correct answer. So A is 3. The only option that you're finding here is option number 2. But let's anyway check the rest of the option. The second option, pyruvate. So pyruvate is a precursor which actually gives alanine molecule. And pentose, which is PPP pathway. From there you will actually synthesize nucleotide. The Nova synthesis will be talking about. And oxaloacetate will produce aspartate. So let's talk about the second, third and the fourth ones. How a pyruvate is going to give a alanine molecule. So we know a pyruvate is going to be a 3 carbon compound. CH3, C double bond O and COO minus, which is a 3 carbon compound, which combines with one of the amino acids, which is acetic amino acids like glutamate. So this is going to combine with a glutamate molecule. And it's going to be a 5 carbon compound. 2, 3, 4 and 5. And COO minus is here. COO minus. And then here onto the second carbon, they have amine group. And rest of the things you just have to valence it. So now this enzyme is definitely going to be an important enzyme, which is alanine transaminases, or you can say aminotransferases. Alanine transaminases. And what exactly happens is this amine group will be given to this second carbon of the pyruvate. There is just the transamination reaction takes place. So now it becomes what? It becomes CH3, C, here the amine group comes. So which becomes NH3 plus and COO minus. And now we call this as alanine, which is L-alanine initially and then becomes a D-alanine. And the rest of the things, the aldehyde group, ketone group exactly. So this ketone group actually comes over here onto the second position. So there is just a shifting of this amine group. So transamination reaction is taking place. So the rest of the things remains the same. And this molecule is called as alpha ketoglutarate. So definitely a pyruvic acid is giving, it is acting as a precursor to produce alanine molecule. Yes. What about the third one? So let's look in for the question. The third is pentose phosphate. Yes, we know PPP pathway is definitely going to produce ribose and xybulose. So definitely they are going to make ATP molecule, DATP and all those things. The last one, oxaloacetate to aspartate. That is also a transamination reaction. So let's talk about that also in detail. So let's write oxaloacetate Oxaloacetate is actually a 4 carbon. This is a 4 carbon. This also combines with one of the amino acids, same glutamate, which is a 5 carbon. With the help, let's write the structure 1, 2, 3 and 4. And here it's definitely going to have an acid group. And here the glutamate, we already know the structure, it's 5 carbon. Here they have an amine group. The rest of the things remains the same. And here also the same thing will happen, which means there is going to be a transfer of this amine group to here and this ketone group to here. So now with the help of which enzyme? Aspartate transaminases or aminotransferases. Aminases AST. It is going to be converted into, this becomes which molecule? This is very important, NH3 plus, and this is going to be CH2 COO minus, and this molecule is going to be an aspartate. So who is actually giving an aspartate? The rest becomes alpha ketoglutarate, which we have already seen. So oxaloacetate is going to be a precursor for aspartate molecule. So if we have to go for the question, we know that hexose is giving a cellulose, pyruvate into an alanine, pentose phosphate to a nucleotide, and oxaloacetate to aspartate. So the correct answer for this question is option number 2. Yes, the same, pyruvate is the precursor, definitely, and because of PRPP, we know that there is nucleotide productions, and glutamate, everything we have seen. The next question, another question is part C question. It is from question number ID is 82. It is a question from the plant physiology only, very specifically from Arabidopsis. Long day plants. They were actually talking about long day plants. They were talking that if there is going to be a plant which is exposed to light, will it show seedling and flowering? That's going to be the question. So if we have to talk about this in detail, we know that there is something called as etiolated seeds and there is something called as light exposed seeds. So to make it very simple, I'll explain the concept and then I'll explain the answer exactly. So if the plant is actually kept in a dark situation, let's take Arabidopsis long day plant, and their seed is kept in a dark situation and one is kept in a light situation. So if I'm going to keep this in a dark situation and grow this, then these seeds are called as etiolated seeds. If I'm going to keep it in a light situation, then they are photomorphogenic seeds. So here they are exactly talking about photomorphogenesis. Genic seed here. So this light conditions they are talking about. So you should understand the point that they are talking about photomorphogenesis. So now we have to understand if it is kept in a dark conditions, what are the things that can happen? So let's consider this is the root. The root will not be well developed. This is the root. Root not well developed. And they have a very long or elongated hypocotyl. And they have this apex part, or we can say this apical hook. This is apical hook, and they are closed. And they do not have unexpanded cotyledon. Cotyledons are not expanded. But if light is present, everything will be just opposite. The root is very, very well developed. And the hypocotyl becomes short hypocotyl. And the cotyledons are really, really expanded and they are going to be green in color because the light is there. So they synthesize chlorophyll. So the cotyledons are actually expanded and there is no apical hook and they are open. So now the situation is when I'm going to expose this Arabidopsis seeds in a light conditions. These light conditions are actually induced by some photoreceptors which are present in the plant. The one which actually receives light. There are some photoreceptors in plant. So those photoreceptors, we know one among them is phytochromes. And this phytochromes, actually we know it's a dimer which is usually observed two important lights. One is going to be a red light It receives red light. Another one is going to be a far red light. We know that a red light usually will be in the wavelength of 630 nanometer and far red light will be in the terms of 730 nanometer, 720 to 730 nanometer it will be. So what will happen? So if a phytochrome observes red light, it is called PR. If the phytochrome observes far red light at a wavelength of 720 nanometer, it is called PFR. So which means we can say a phytochrome exists in two forms, PR and PFR. Just remember, PR is inactive, whereas PFR is active. So we know that if you want seedling, the question is seedling, if you want seedling, germination and then stem development and flower development to take place, you definitely want light. But when you expose red light, what will happen? When you expose far light, what will happen? When you keep them in a dark light, what will happen? You already know when there is a dark phase, nothing can be done. So we can say that this cannot be the answer. Let's make everything clear. So we know that PR, phytochrome PR is actually an inactive form, whereas PFR is going to be an active form. Which means whenever this phytochrome which receives a red light, when it receives this red light, it will be converted into PFR, which is active. Okay, but when PFR is absorbing far red light, because PFR can absorb only far red light, when it absorbs far red light, it will be converted into an inactive form. And the same PFR will be converted into PR, inactive form, during dark situation also. This is the slow phase. PFR to PR is a slow phase. So now let's check the statements. Far red light followed by red light. What does it mean? So who will observe far red light? PFR. So PFR receives far red light. Far red light, which means an active form will be converted into an inactive form. Okay, so far right lay are getting it. PFR receives a far right red light and it will be converted into PR inactive followed by red light. Now PR will receive red light and now it will be converted again into what? PFR, which means it is going to be active. So the first statement goes correct. If it is active, it will induce seedling, it will induce flowering, everything is possible. So the first statement goes correct. So you can eliminate option B and C. So we are left with only two options. Okay, let's check. Second option, far red light, PFR, which is active form, is going to absorb far red light. If they absorb far red light, they will be converted into PR. Now PR absorbs inactive, absorbs red light. It will become what? PR absorbs red light, it will become PFR. But then again it's going to be a dark phase. No, it will not show any kind of thing. So you cannot choose option number B. So you can see this option also goes wrong. B is there. So definitely the option is going to be D. But let's check the rest of the options. So the third option is red light. So who will observe red light? PR. So PR is inactive. So PR is going to receive red light and now it will get converted into far red light, which is PFR. Now PFR will absorb which light? Far red light. Far red light. If they absorb far red light, it will again get converted into PR, which is inactive. If they are inactive, no seedling. Or we can say there is no flowering happening. So this statement goes wrong. So we can choose only first statement. Second statement is wrong. Third statement, yes, dark phase. So okay, there is a ethylated seed. I am taking that ethylated seed which is grown in the dark condition. And now I am exposing them to far red light. Who will observe far red light? PFR will observe far red light and convert into PR, which is inactive. Now this inactive form receives a red light and now it will be converted into PFR, correct? So which is active. So now this active form will show what? Yes, definitely a flowering. So D option is correct. Yes, so first option A is correct, D option is correct and the last option is E. So far red light followed by dark phase. No, in between if dark phase come, nothing can happen. Then red light. Red light means you should remember it will be converted into PFR. Yes, PFR. This is a correct statement actually. So option E is a correct statement. Option F is a wrong statement. So let's check for option number E. So here what they are telling, far red light. So PFR observes far red light, active form, into a dark phase. Okay, and then this, when ethylated seed is given red light, who will observe red light? PR, and it becomes PFR, which means they are active. So option E and option D and option A. So the correct answer is going to be option 4. Okay. So the next one is part C question, which is from plant hormones. Easy question. So following are certain statements regarding gibberellic acid signal transduction, signaling pathway they are talking. DELA proteins, which is a repressor. Remember, DELA protein is a repressor. Repressor protein. DELA means D is going to be aspartate, glutamate, leucine and alanine protein, which has N terminals, all these amino acids, and they have GRAS domain in the C terminals. So negatively regulate G signaling. Yes, when there is a repressor, definitely it will not allow any kind of signaling. So this statement goes correct. So where are you seeing A? Yes. So we will eliminate this option and this option. So we have to find out whether it's 1 and D. So let's understand degradation of GA receptor is mediated by DELA proteins. Let's understand the pathway and then come here. Okay, let's go for this. Suppose if I have to talk about GA. GA active form is GA1. GA1 is active form. So now what exactly happened? GA3 is also active form. Suppose if GA comes and binds to this receptor. Suppose we know that GA is responsible for what? Seed germination. If I want a seed to germinate, first the seed will actually imbibe water. And as they imbibe water, the embryo is going to synthesize GA1 active forms in the plastids, endoplasmic reticulum and in the cytosol. And now we have this GA present. And now this GA come to the amylose. Suppose let me make this as a monocot seed. So this monocot seed, the water is actually imbibing. So here we have the aileron layer of the monocot. So this is the aileron layer. And inside you have aileron layer and inside you have an embryo. So this is going to be the embryo which is actually present. So now what exactly happened? The water enters here. As soon as the water enters here, the embryo is going to synthesize, which means every cell in the embryo is actually going to synthesize GA1. So in this cell, what exactly is happening? In the aileron layer. So this GA1 will be transported to which layer? Aileron layer. So in aileron layer, there are a lot of cells. So this GA will enter into every cell and what's going to happen? So they are writing this is the aileron layer. So here this GA comes and binds to a receptor called GID1. This is the receptor. So this receptor is called gibberellic acid insensitive DARF receptor. DARF receptor. As soon as the gibberellic acid comes and binds to the receptor, this complex is activated. So as soon as this complex is activated, it is going to activate G-coupled proteins, which is heterotrimeric protein, which means alpha, beta and gamma, 3 different groups are present. To the alpha, GTP is attached. Now we have a substrate. Now we need an enzyme. Now they will activate one enzyme called guanyl cyclases. And an enzyme is present, substrate is present. So they will combine together, make up CGMP. Yes. Now this CGMP, calcium independent pathway actually, this CGMP is a secondary messenger. Now it will send a signal and produces an intermediate molecule which is called E3 ubiquitinases. Because this ubiquitinases is going to be a complex, SEF complex, which has F box very specifically. And this F box is definitely going to recruit one protein to degrade them. Okay. So what is that protein? That protein is going to be DELA. We have to degrade this DELA. So whenever a GA comes and binds to the GA receptor, then this intermediate is activated. Then this intermediate will go and take up the DELA repressor. This DELA repressor is actually present in the nucleus. You can see. So this is E3 ubiquitinases. It enters inside the nucleus. And now it will understand, yes, I have to degrade this one. And it will add ubiquitin to this along with 26S proteasome, and they will degrade this. As soon as a repressor is degraded, yes, the transcription can take place. GAMYB gene will be transcribed. So we have mRNA. This mRNA comes out. Now this will make a protein, which is GAMYB protein, which is a transcription factor. This will again enter inside a nucleus, and then it will allow the synthesis of alpha amylases. And this alpha amylase gene will produce mRNA and then alpha amylase protein. Why do we need this alpha amylase? This is in the aileron layer. So this is the monocot seed, and every synthesis is now, or the signaling is actually taking place in the aileron layer. Now we have synthesized alpha amylase in the aileron layer. But this is the endosperm. And in endosperm we have what? Starch. So if you want the starch to break, you need alpha amylases. Now we have synthesized alpha. So from aileron layer it will go to the endosperm. And now it will break all the starch into glucose. And now this glucose will convert into ATP. Yes. Now if ATP is there, definitely seed germination will take place. So we can say gibberellic acid is responsible for seed germination. Now let's understand the options. DELA protein negatively regulate. Yes. If DELA protein is present here, it will not allow this transcription. So we have to degrade this. So this has to be produced. When this will be produced? Only when GA and GA receptors are bound together. So let's check. Degradation of GA receptor is mediated by DELA protein. No. GA receptor, nothing will be degraded here. So this statement is, DELA is not coming here. DELA is actually in the nucleus. So nothing to do here. This is a wrong statement. The next one, ubiquitination and subsequent degradation of DELA protein. Who is degrading it? E3. So E3 is coming here, and this E3 is degrading this DELA protein. Is actually independent of GI1. No, only when GA comes and binds to GI1, they produces E3. Only if E3 is produced, ubiquitinases, they can degrade DELA protein, not independent. So this statement is also going to be a wrong statement. Okay, last one. GA binds to GID1, promotes binding of GID1, which means E3 is produced, and then to the DELA protein by adding up ubiquitins and then degrades it. So D and A are the correct statements. So which of the following is correct? Definitely A and D. So the correct answer is option number 4. The last question is question number 85, which is part C. So you can see here there are different kind of reactive oxygen species generated in the plant. The first is H2O2 is relatively more stable. As we know, H2O2, superoxides, hydrogen peroxides are all reactive oxygen species. They are very stable. Yes, when you talk about all the reactive species, they are more stable and they are membrane permeable and they can travel long distance. Correct. We have to choose the correct statement. ROS is scavenged by only through enzymatic reaction. Only is the word here. It is not only by enzymatic reaction. You can see. It is not by enzymes like ascorbate peroxidases or superoxides or catalases. Even non-enzymatic reaction can also be scavenging this ROS. So this statement is wrong. Not only enzymatic, even non-enzymatic reaction. Ascorbate glutathione cycle is associated with scavenging ROS. Yes. This cycle always removes ROS from this one, which causes unstable conditions. Next option is monodehydroascorbate reductase is not an antioxidant enzymes. So which means they are talking about this is not an antioxidant enzyme. It is an antioxidant enzyme. They are telling no, it's not an antioxidant. No, it is actually an antioxidant enzyme. So this statement is also given wrong. So we can say the correct option is one, which is A and C, which means A and C is the correct one. The second and the fourth is the wrong one. So we can say A and C. The first option goes correct. The last question is question number 86. Very easy question. They are talking about apoplast, symplast and transcellular transport. Let's check this one. We know that this is apoplast. Apoplast usually goes through the cell wall. So you can see here this violet part. This apoplast is actually going through the cell wall. Here it is going through the cell wall to the epidermis and then to the cortex and then to the endodermis. But in endodermis they have Kashperian strip which is made up of suberin, so they cannot cross. Then they come to the symplast pathway and then go to the apoplast. So this is the apoplast pathway which goes through the cell wall. In Simplas, this green part, if you see, it goes through the cytoplasm of every cell and go through the plasmodesmata. So Simplas goes through the plasmodesmata and cytoplasm. But transmembrane will go to the vacuoles. You can see this is white part is the vacuole. This is vacuole transmembrane transport. So it is going through the vacuole and reaches the xylem, like going through epidermis, cortex, endodermis, pericycle. So you have to remember apoplast is through cell wall, Simplas is through cytoplasm and plasmodesmata, transmembrane is through vacuoles and plasmodesmata. So remember this is through vacuoles and plasmodesmata, which is a connection between two cells, and Simplas is through cytoplasm and plasmodesmata. So now let's go for the options. Very easy options, you can see. Apoplast transport is mainly responsible for which of these things? Apoplast transport goes through cell wall. Yes, I told you apoplast will be through the cell wall. We said it is through cell wall, correct? And the next one, apoplast transport mostly involve plasmodesmata. No, you can see it is not going through the plasmodesmata actually. It is going only through the cell wall. So this statement goes wrong. But Simplas predominantly occurs through plasmodesmata, correct, because cytoplasm and plasmodesmata. So this is a correct statement. And transcellular transport goes through vacuoles through tonoplast. Yes. So it is going through vacuoles and this is the tonoplast. So it is going through the plasmodesmata and vacuole tonoplast. So this statement also goes correct. So what are the statements correct? A, C and D. So the correct answer for this question is option number C. So in today's video, we were actually talking about all the questions from CSIR NET morning shift, very specifically 2022. So I believe that this video is helpful for you. So I'm going to meet you back again with yet another wonderful video. Thank you all of you.